There's the recording. Share my screen. There we go. I turned on the light too. It's starting to get dark in here. Of course, with this green shield here, you're not seeing too much light. You don't see the green shield behind me because it's supposed to show this picture there. That's why I have the green shield behind me. But, let's see. I'm just rambling, aren't I? All right. So we're going to begin lab 2 tonight, aseptic transfers. You should also have had read the textbook chapter 3 through the end of the section, the compound light microscope. And you'll need to read that before lab 3 and lab 4. So try and read the textbook in chapter 3 beforehand. I also need to begin and discuss the infectious disease project. So let me do that now. Sorry, we have a lot to do today in the lab. So in the infectious disease project, come down to, I think it's week 2 lab. You are seeing my screen, right?
Yeah.
Right, infectious disease research project. It's not due until November 6th, but you need to get started on this research project. It will take you time. This project is worth 100 points. Yep, lost that. Let's get rid of that one if I can. Come on. All right, let me see if I can move that. There we go. Am I there? I am here, all right. So when you come to this page, you need to download the infectious disease project. I think I will download it because it'll show better as a download. And eventually you upload it. The document has to be a Microsoft Word document or a docx document or a rich text file, formatted file, or a PDF file. Some people have been trying to send me, I don't know, documents, and they're not sending it to my site here, probably because it's not allowing them to download it. It's because they have the wrong file type. I tell you right here what file types I accept. And then they sent me this file that I couldn't open. And so you need to pay attention to the file types that I have here, because these I can open. Let's see, where is it? I'm not seeing it. Yeah, I did press that, right? And something happened, right? I hate to do it again. It looks like I'm going to have to. Let me, nope, I'm not seeing it. All right, I'm not sure why that's not opening. Let me come to my version of it. I wonder if it's there. Nope. Check in to see it's green. Oh, there it is. For some reason, didn't see it. I think it just was opening. There's probably going to be two of these. All right. Put that over there. Your attention is going to be focusing on a specific infectious disease with the goal of determining the underlying cause of the disease, what the microbe does to the human body, how we treat the disease, and how social and economic and political factors affect our ability to deal with the disease. You can read about the rationale. The format, what you need to do is pick an infectious disease and then send your request to me. You should have maybe 2 or 3 possibilities in case your choice is already chosen, meaning only one person can have one infectious disease in the class. I know that 2 people are interested in AIDS. So if you're interested in AIDS, you better get it into me right away, because the first person who gets it, gets it. If no one has your disease, I will then tell you that it's fine, or I will tell you that you need to narrow it down or pick another disease. So you need to email me and get it approved. I'm just going to be checking it. Occasionally, like some person, I think it was last term, but in the future, in the past terms, this one is frequently requested, and they say, I would like to do it on pneumonia. And I'll tell you, no, you can't do it on pneumonia, because pneumonia is a disease that is caused by multiple organisms. And that would be too broad of a topic for this project. However, I mentioned that they can pick one specific organism that causes pneumonia, like Streptococcus pneumonia or Klebsiella pneumonia, and then write it on that organism that causes pneumonia. Is that clear? So you need to pick one specific organism that causes one specific disease. And for some of them, like pneumonia, there's multiple diseases or disease-causing organisms that can cause that disease. And you have to pick one organism. I think there's a fungus that can cause it, and then there's viral pneumonia as well. So like I said, you have to pick one. Anyways, that's what I am. I'm sort of a gate checker. I'll also tell you, sorry, that topic has already been chosen. So like I said, have a backup, at least one backup, in case your topic is not allowed. Very rarely do I tell people, you're not going to be able to do it on that disease because it's not an infectious disease. So it has to be an infectious disease. And there are a few diseases which are not really infectious, but we call them infectious diseases. And let me see, botulism is one. You can do.
What about flesh-eating disease?
Well, as long as it's contagious, it would be infectious. If it's caused by a microorganism, it would be an infectious disease. Botulism, you can get it from eating contaminated food, especially canned food, okay? And it's not infectious once you get it, but to you, it's infectious. Does that make sense?
Mm-hmm.
Okay. But somebody did once, like, I want to do it on lung cancer or something like that. And I tell you, no, you can't do it on that. It's not an infectious disease. Okay.
I just had a quick question.
Once did diabetes, and generally diabetes is not an infectious disease. Yes, what's your question?
So are you now accepting like topics for the infectious disease? So that's like ready that we can start emailing you now?
Yes, okay. You can send it in now. Okay. I am now ready to receive those. Try and send them to my Canvas website. I'll be checking that one first. So if you have it there, you'll get first choice in case somebody else requests it. I try not to use my what is it, clark.edu campus mail very much, especially with students, because I get a whole lot of mail from my clark.edu mail site, and I just, most of it is just garbage to me, so I don't check it as often. All right, you need to do a literature search. Most of that will be online, but make sure you have valid sites if you go online. An easy way to check if you have a valid site is just make sure that the 2 or more websites are in agreement. If assuming they're, pseudo valid sites, because if you go to a site and you're going and you're getting political information, or a political site, it may not be valid scientifically, and that should be apparent if you go to a valid scientific site. If you go to another political site, it may have the same information, but it may not be scientifically valid, meaning it may be garbage. So try and use valid sites. You do need to have valid references. Since I brought that up, let me check on that right now. The sources you cite in your paper should be scientifically appropriate and reputable. Those include government information, scientific journals, popular magazines that are known to be reliable. Books and textbooks are actually good sources. So actually your microbiology textbook is acceptable. If you don't know, all scientific textbooks are reviewed by other scientists or experts. So it could be newspapers of a credible source like the New York Times is acceptable. The National Enquirer and the Sun are not acceptable. Scientific television shows are acceptable. Sources to be suspicious of include religious, political, privately funded publications. They're going to be slanting things to support their view. So be careful of those. It's best not to use them. Personal websites, you shouldn't use those either because anyone can put up anything on the internet. College and university websites that have pages that are made by a student should not be used. If it's a college or university site that's put up by a professor, it's probably reliable. Wikipedia, you can use it as your initial source, but don't give it as a reference. Because the trouble with Wikipedia is anybody can go in there at any time and then change the information. And because of that, it's not considered a reliable source. In reality, Wikipedia is pretty reliable, but because anybody can change it and then put in garbage, we won't allow it as a reference. But what you can do is use Wikipedia and then get a reference to back up what Wikipedia is saying. Any questions about any of that? All right. Some things that are required for this, you must have a cover information that gives the title and the name of the disease, your name, the date, the course name, that's Biology 260, Microbiology, but don't make it a cover page. Put it on the top of the first page of your text. If I print it out, I do not want to print out a cover page. Is that clear? You'll actually get 1 point if you have the cover information on your text page, assuming you have all the information in your cover page, or cover information, okay? If you have a separate cover page, the most you're gonna get is a half a point, all right? The text, that'll be worth 60 points. The material in your text should be in a readable, grammatically correct, spelled correct. It should flow reasonably and be readable. As much as possible, it should be written in your own words. Whenever you are not using your own words, it has to be in quotes, and then you state where you're getting it from. Wherever you have a quote, you should say where you got that quote from. And wherever you're using information that's not common knowledge, you have to give a in-text quote. I'll talk about what that is in a moment. Like, for example, if you're saying that COVID-19 is currently a pandemic, that's common knowledge. You don't need to have the information. But if you were to say something like, I don't know what the death rate is in the state of Washington, we're just going to say the death rate in the state of Washington passed 10,000. Okay, yesterday, and give the date. I don't know what the death rate is, but I'm just stating if you give a fact like that, you have to give a quote because most people don't know that information. An in-text quote. I mean, an in-text citation, sorry. Is that clear? Wherever you're giving knowledge that's not common knowledge, give an in-text citation. Okay, if you don't have enough in-text citations, you'll get a deduction. Generally, as long as you're fairly good, I won't mark you down on that. But if you really have very few in-text citations, you'll get a deduction. It's kind of hard to have too many in-text citations, but if you find that you're giving an in-text citation after every sentence, what you should probably do is wait until the end of the paragraph and then just give 1 or 2 or 3 in-text citations at the end of the paragraph. Any question about that? Okay. All right, an in-text citation must be in the correct format. It's going to be worth 10 points. Normally, what you're going to be doing for the in-text citation is you'll say something in your body of your text. There were 10,000 deaths in the state of Washington as of September 28th, 2021. And don't quote me on that. I don't know what the numbers are. I haven't looked it up recently. Okay, but you should then put it in parentheses and then the author's name, who you're citing, it's the last author's name. So for me, that would be Volpe. And then the year, 2021. If it's 2 authors, you say Volpe and Smith, comma, comma, 2001, and then in the parentheses. If it's more than 2 authors, you say Volpe et al, comma, 2001 or 2021, and then the parentheses. So that's normally how you do an in-text citation. If you don't have the correct format, you'll get a slight deduction, but at least I know you're trying, so it won't be a major deduction. The in-text citations are worth 10 points. It has to be used in a commonly used citation format. I don't care if it's APA, MLA, a scientific format. Just be consistent, meaning you're using the same format throughout your entire paper. Any question about in-text citations? All of your in-text citations, which are in the text, should then be followed with the formal citation. And I'm going to say at the end of the paper, you have your references. That's your formal references, where you give more information than when you have in the in-text citation. It's called the literature citation page, a list of all the sources cited within your text. You should give here all of the authors' names, as well as their last name and then their first name, or at least their initials, and then the year of publication, the location of the article you got, so I guess you'd say the title of the article, and then the location of the article. If it's a scientific journal, you'd say something like the Journal of the National Academy of Sciences, and then the volume number, and then the page number. If it's a book, it's a little different. You give the article that you're quoting, and then you give the book name, and then you give the editors of the book, the publisher of the book, and the year the book was published. Oh, and you give the city of publication as well. Okay, if it's a web page, what you're doing there is give the author's name, the year of publication, and that one might be a little tricky. Go dig around looking for it. If you can't find it, use the format it says to use in the whatever format you're using, and it's slightly different for APA and MLA. I think one of them, it tells you no year, others you just leave it blank. You have to look up how to do that depending on what format you're using. And then you give the location of the web page. For example, this one would be https://clarkcollegeinstructure.com, etc. And what the best thing to do is just copy and paste it. Okay. Any question about that? All right, so you know how to do both in-text citations and literature citation page. These are the most common errors that students make. That's why I'm taking time to tell you how to do these. Go through what else has to be in the text. I don't have it there. Let me look down further because I've just got the text. I don't say what the text has to be. All right, so then there's the literature citation page. That's worth 15 points. In the body of your text, you should have, this isn't in there, but I should put that in there, but I've got it up here, but I should have it down here. You should have the cover information, which is on the same page as your first page of the text. You should also then have the causative agent and put that in, what do you call that, a subheading? And then you have to talk about the causative agent, and it depends what the causative agent is. I'll let you read about that. And then you should talk about the history of the disease or the causative agent. How long have we known about the disease historically? How was the disease treated? Is there any history of vaccine development? Describe the changes in our knowledge and attitudes towards this disease throughout history. Usually students are pretty good about this, but occasionally they don't talk about how our attitudes have changed towards this disease. Now, if it's something brand new like COVID-19, there hasn't been a whole lot of change in our attitude and knowledge. You could say that initially people were very fearful and we tended to shut down. And nowadays we're not going to be doing that. And the trouble is people have lost their fear of it and they're going around picking it up and spreading it around, which is a bad thing. Okay, that could be described. But generally there won't be a lot of history in the changes in knowledge and attitudes about COVID-19 because it hasn't been around for very long. But if you're talking about a disease which is ancient, like trying to remember the name of that disease. Geez. It's known in biblical times and it's contagious, spread from one person to another, and it caused the flesh to rot.
Leprosy.
Thank you, leprosy. That one has changed dramatically because it's been around for so long. Initially, people thought they were unclean and they would say unclean, unclean to warn people that they're coming next to a leper. And now we know that actually the disease causing leprosy, in the first place, is not very communicable. And only, I think, like 10 to 15% at most of the people can even contract it. You have to be susceptible to it before you can get leprosy. Okay. And they used to have colonies, and they had colonies where they would put the lepers, get them away from society and keep them in their own colony. And nowadays, of course, leprosy can be treated with antibiotics. And so if somebody does have leprosy, they will be treated, and they're treated at home, and they continue their work and all that other stuff. Okay. Anyways, talk about the history, talk about the epidemiology, talk about the pathology, talk about the response and the treatment towards the disease. And then lastly, the socio and political and economic considerations. So I have a subheading for each of these and then talk about that. You'll note that there's 1, 2, 3, 4, 5, 6 topics you need to discuss. That means each of them should be less than a page, because I'm asking you to make this into 5 or 6 pages. And that includes the references at the end. Okay? Any question about any of that? And then at the end of this document, I've got the grading, how you'll be graded. 1 point on the cover information, and make sure it's at the top of the first page. The causative agent's worth 9 points. The history is worth 10 points. Epidemiology, 10 points. Pathology, 10 points. Response and treatment, 10 points. Socio, political, and economic issues, 10 points. The quality of writing, the grammar, the spelling, the punctuation, the flow, the readability, 15 points. In-text citations, 10 points. Literature citation, 15 points, for a total of 100 points. Any question on this project? All right, so pick a topic. It's probably best to have a backup, send it to me, and then I'll get back to you and let you know if you can have that topic. Okay, each student has to have a different topic from all other students. That's my only concern there. Okay. All right. But I would allow like different people to have like streptococcus pneumonia, Klebsiella pneumonia. I'm trying to remember another one that causes pneumonia. There's several others that cause pneumonia. Okay. All of those would be valid. Okay.
And you said we can start emailing you those now?
Yes.
Okay.
If you wanted to do it in the lecture, I suppose that's fine. Okay. All right.
Hey, I have a quick question. Do you have like a total amount of words that we're supposed to have, or is it just 5, 6 pages?
No, 5 or 6 pages. You can make it single spaced. If it's a little bit long, you're not going to get a deduction. Like 7 pages, I won't deduct it. But if you're 10 pages, you'll probably get a slight deduction. Okay. If it's less than 5 pages, you'll also get a deduction.
Okay,
All right.
perfect. Thank you.
All right. And that can be single spaced or double spaced. I don't care. Do try and make it. I've got it in the directions. Make it a 12-point font. As you can see, my eyesight isn't as good as it used to be. So make a 12-point font. 10-point font is just too small for me to read. All right. Any other questions? All right. Let's go and talk about the lab. I had it open at one time, but I think I moved it around looking for the in-text file. Module 2, there's the teacher's copy and there's worksheet 2. Let's try putting that here. So upon successful completion of this lab, students should be able to understand the purpose of using aseptic transfer techniques, determine if aseptic transfer was used from the data provided, and define the following terms: ubiquitous, aseptic transfer, inoculation, and contaminant. In our local environment, microorganisms are ubiquitous. This is problematic when working in a microbiology lab because we generally want to work with a pure culture, a culture that contains one and only one species of organisms. Louis Pasteur developed the concept of a pure culture and then the technique for getting a pure culture using aseptic transfers to minimize the risk of culture contamination. So we're going to learn aseptic technique today. We'll learn how to make an aseptic transfer. So follow the lab protocols. During inoculation, never lay the test tube cap or the Petri dish lids on the workbench, because when you do, let me see if you can see this. We're going to pretend this is a cap. When you lay it on the workbench, things from the air can drop down and then contaminate the cap. And then when you go and put the cap over the dish, they'll fall off the cap and then contaminate your dish. So don't lay things down. What you should do is hold them in your hand and then work with it in your hand. Another thing, when you're doing a dish, you should take the lid and get it up high enough so that you can streak or do something on the plate, but leave the dish so it's largely covering, or the cover of the dish, leave it on top of the dish so that if anything falls down from the air, it's likely to hit the dish and then fall off of the cover of the dish as opposed to falling in the dish. Now, obviously you will have a little bit of the dish open and risk contamination, and you have to have it open so that you can work. Okay, any question about that? Work efficiently, try not to talk while you're doing transfers and working with the inoculation. You need to sterilize the loop and the needle or the forceps or your other tools beforehand and after they are used. You do it beforehand because if you don't, and then you put in bacteria, you're going to put in contaminants as well. And you do it afterwards because you don't want to take this bacteria and then put it on your counter. The counter will be covered with bacteria. Any questions about any of that? In order to inoculate, that means transfer cells into a new sterile media, we just make sure to sterilize everything that can potentially contaminate the sterile media. If you were to be in the lab, we'd have a bacterial incinerator, and you put the loop or other tool like this, where the metal portion is put in the bacterial incinerator when it's red hot. You could use a flame, but we don't use that anymore in the lab. Know the terms of this lab. I'm not going to go over them. I'll let you read them. And then do the laboratory exercises. We're not going to be physically performing these activities, but you will be watching videos of these activities. We expect that you will know and understand the material in these videos and in this lab manual. Procedure 1, you're going to transfer a sterile liquid using a glass serological pipette. In procedure 2, you're going to transfer a sterile paper disc using sterile forceps. Please watch the following video demonstrating proper aseptic technique. Okay? In procedure 1, you're going to be transferring 1 ml of sterile water using a sterile pipette and put that 1 ml into nutrient broth. When you're reading the pipette, it's important to realize that the liquid will not be straight across. It'll form a meniscus, and how you read the meniscus is not at the top, not in the average or in the middle. You read the meniscus at the bottom of the meniscus. Any questions about that? Let's scroll down and come back to procedure 2. Now let's come back to procedure 3 and 4. So we got pictures for procedure 1. Right after here. There is procedure 1. This tube here, that's what P1 stands for, procedure 1. And you look at the picture and you determine, is it cloudy or is it see-through? And then you come up here to the table to help you understand the results for procedures 1 and 2. If the nutrient broth is clear, there's no organisms growing in the broth, aseptic transfer was successful. If the nutrient broth is turbid, that's hazy or cloudy, organisms are growing in the broth and aseptic transfer was unsuccessful. Any question about that? And then you answer the questions right here. We'll come back to the questions in a minute. Let's go back to procedure 2. Procedure 2, you're going to use aseptic technique to transfer a sterile paper disc into the nutrient broth. Remember, a broth is a liquid culture. Label the nutrient broth, use sterile forceps to transfer the sterile paper disc into the nutrient broth. Work quickly so that it doesn't get contaminated from the air. Replace the lid with the nutrient broth. Incubate the nutrient broth. Scroll down here. In this table, it gives you the results and the conclusions. It's the same for procedures 1 and 2. And then this is procedure 2's tube right there. And you see that white down there? That's the paper disc you put in. Okay? Any questions about procedures 1 and 2? All right. For procedures 3 and 4, we're going to be transferring Serratia marcens to a broth. Serratia marcens is a bacteria, and we're using this one because it's red colored. In procedure 4, we're going to be transferring Serratia marcens to an agar slant. Remember, a broth is liquid, an agar slant is a solid tube. For procedures 3 and 4, watch this following video, demonstrate how to correctly transfer from a bacterial stock culture to sterile broth and sterile agar slant. There's some important notes about this video. You only need to watch until 4 minutes and 23 seconds. The bacteria being transferred in the video is E. coli. And so the bacteria in the video will be white. And we're telling you the bacteria Serratia marcens is red. So realize that we don't have a video showing you Serratia marcens. And then there's an error in the video. When the instructor puts the loop in the E. coli stock tube, you can hear something. It's between 3 minutes and 5 seconds and 3 minutes and 20 seconds. Did you hear that? Do you know what that error is? You can hear the sizzling, okay? And I think there's a question down here about that. So for procedure 3, you're transferring Serratia marcens into a sterile broth. When Serratia marcens, which is red, is growing in the broth, it'll appear pink. You should understand that when you have a broth culture, all the bacteria grow together. If you have a major contamination and you're using Serratia marcens, the color may not be pink, okay? But if it's a minor contamination, the Serratia marcens will make it look pink. And so if there's minor contamination, you can't tell whether you got a minor contamination because the bacteria will just all grow all together mixed up in the broth. And then this is the procedure for growing the broth. Read through that. and then come down here and look at the results and interpretation. If the nutrient broth is clear, no organisms are growing in the broth, so aseptic transfer was unsuccessful because we're trying to transfer Serratia marcescens into the tube. If the nutrient broth is hazy or cloudy, organisms are growing in the broth, and you can assume aseptic transfer was successful. And then come down to the picture, which is P3 right here, to see what happened. And then let me move on to procedure 4. Here you're transferring Serratia marcescens to an auger slant. If the Serratia marcescens grows on the auger slant, it'll grow red in color. Contamination is more easily detected on a slant than in a broth, because what you're going to do is there's the slant there. You're going to streak it on the slant. And if the bacteria is only growing on the slant and is red, then it's good. And if one area, like right here, or maybe right here, is white or some other color than red, then you know you have contamination. Any question about that? All right, read through the procedure for procedure 4. Look at the results and the conclusions. If the slant does not show confluent or colony growth, no organisms are growing on the slant, aseptic transfer was unsuccessful. If the slant does show confluent or colony growth, then organisms are growing on the slant and aseptic transfer was successful. You can also look to see if it looks red and if there's any contamination in the area. Right there, that's not contamination. That's just where a drop of water is on the bottom. Okay. And then answer the questions down here. This question 0, which you'll have it labeled 0 in yours, does need to be answered. And then answer question 1 about procedure 1. Answer all the questions. Then there's for procedure 2, question 2. For procedure 3, question 3. For procedure 4, question 4. Answer the questions. You don't need to do the references, and then submit it in the lab worksheet for lab 2. Any questions about any of that? All right, if there's no questions, go ahead and get started on the lab. I will be here until 8 o'clock or the last student logs out. It's now 7:16, and I think everyone will be done by 8 o'clock. If there are any questions, I will answer questions for you during the lab. Okay? All right.